Hi everyone, welcome back to my channel, and today I'll be talking about the foundations of retroviruses. What are retroviruses? So essentially, retroviruses are basically a type of RNA virus that infects host cells to survive and reproduce. And since they can't really do that on their own, they have to rely on cells that they can infect, which we call host cells. They're comprised of RNA, a protein capsid, a lipid envelope, and viral enzymes. One of the most important enzymes that they have is called reverse transcriptase, which I'll go over later. Essentially, reverse transcriptase allows RNA to convert into DNA. Then that DNA becomes a part of the host cell's genome, and once that occurs, the virus can use the host cell to create more retroviruses. An example of a retrovirus is HIV, which I'm sure you guys have heard of before. And basically, HIV mainly infects our immune cells called the CD4 T cells. These are really important because they help our body fight off infections. And HIV can be transmitted through bodily fluids, including sexual contact, sharing of needles, and also even childbirth. Over time, as the HIV damages more and more of our immune cells, our immune systems get weaker. The good thing, though, is that HIV can be treated with antiretroviral therapy, or shortened as ART, and then this helps lower the amount of virus in the body so that people can manage it much better. First of all, retroviruses are RNA viruses. This means that their genetic material is RNA instead of DNA. This is different from humans because we know that our genetic material is DNA. So this is important because RNA carries the viral information, but even though they have the genetic material, they can't reproduce by themselves. This is why they require a host cell, because they don't have all the tools and materials to make more copies of their own. So basically, they depend on the host cell's machinery to do that for them. Like I said in the last slide, the host cells are important to the retroviruses because it provides the materials and energy to keep them going. So basically, once the retrovirus or the virus gets inside, it uses the host cell to copy their genetic material and create new viral proteins. Then those parts can be put together to form new viruses. So for the retrovirus, an infection is good because it helps it going and keep its life cycle continuing. But for host cells, including us, it's going to be harmful because the cell is basically being taken over and used. Now let's talk about the composition of retroviruses. First of all, they have an RNA genome, which is basically their genetic information. Around that, there's a protein capsid. This helps protect that genetic material. You can kind of think of it as like a nucleus in a eukaryotic cell. Around that, there's also a lipid envelope, which helps the virus interact with host cells and enter them. This is also similar to the membrane-bound proteins. They also contain viral enzymes, which are really important because they help with major steps within replication. And all of these parts basically work together to help the virus infect other cells and reproduce. One of the things that makes retroviruses unique is reverse transcriptase, which is an enzyme. The enzyme basically allows the virus to convert RNA to DNA. This is considered unusual in bio because we think of the central dogma where DNA turns into RNA, and which then turns into proteins. But retroviruses basically do the reverse, and that's why they're called retroviruses. This step is pretty important because it lets the viral genetic material eventually become a part of the host cell's DNA. Now let's talk about the basics of the mechanism of reverse transcriptase. So once viral RNA enters the host cell, reverse transcriptase starts by creating a DNA version of that RNA. They basically create a copy of viral DNA. After that, the viral DNA can move toward the nucleus of the host cell. This is really important because it basically sets us up for the next step, which is integration into the host genome. Basically, this means that this mechanism is what allows retroviruses to stay within the cell and continue their life cycle. This integration step is really important because once the viral DNA becomes a part of the host genome, the host cell can start copying and expressing those viral genes that was created within you. So the cell basically begins helping the virus once that occurs. This means that infections within your body can last for a longer period of time instead of disappearing, and because of this, the viral DNA is now a part of your host genome. It can keep directing the production of viral components over time, repeatedly over and over again. Let's talk about a few applications to biology and medicine. Retroviruses are really important in both bio and medicine because they help scientists understand things like gene expression, viral infection, and how genetic material can move within the cells. They also matter within medicine because retroviruses, including HIV, can cause disease. So overall, studying them gives us a better understanding of major molecular biology concepts, and we can use those concepts to connect to different ideas to real human issues. Finally, let's summarize the life cycle of the retrovirus. So first, the retrovirus enters the host cell, then its RNA gets converted into DNA via reverse transcriptase. And then that DNA becomes a part of the host genome, and once it's integrated, the host cell starts producing more viral parts. Those parts then get assembled into new retroviruses or viruses, and then that virus is basically released and it can infect other cells, repeating the cycle over and over again. Thank you guys so much for watching, and comment down below what topic you guys want me to cover next. Thank you.